Alright, so now we're going to talk about staining microscopic specimens. As we mentioned a bit earlier, in their natural state, most of the cells and microorganisms that we observe lack color and contrast. So it makes it really difficult to see things through a microscope, depending on the type of microscope you're using, but a lot of times it makes it very difficult, if not impossible, to see certain things without treating them. So we already alluded to involving different stains and fluorescent dyes, so now we're going to talk about those, very specific ways to prepare things for microscopy. So when we're talking about light microscopy here, we're going to talk about different types of preparation. So remember, we have our light microscopy, we have our EM, our electron microscopy, and then we have our probe. So first we're going to talk about preparing specimens for the light microscopy. In clinical settings, the light microscopes are most commonly used microscopes. So there are two basic types of preparation that are used to view specimens with the light microscope. So we have wet mounts and we have fixed specimens. So the simplest type is the wet mount. The wet mount, in which the specimen is placed on the slide in a drop of liquid. So there's the wet part. You're mounting it on the slide in a wet solution, in water, in liquid. Some specimens, such as a drop of urine, are already in liquid form and can just be placed on the slide using a dropper. Some solid specimens, such as a skin scraping, can be placed on the slide before adding a drop of liquid to prepare the wet mount. So this can come from either something that's already wet, like a drop of urine, for example, or a drop of blood, or something that's dry, and then you just can add water. So sometimes the liquid used is simply water. Often stains are added to enhance the contrast. So you can add water to some of them. Some of them that we use in the lab are going to be just already in a broth, and that will be fine. But other times we'll add stain to it. So once the liquid has been added to the slide, a cover slip is placed on top, and the specimen is ready for examination under the microscope. The second method of preparing is fixation. So the fixing of a sample is referring to the process of attaching the cells to the slide. So this is no longer a wet mount, right? This would be kind of like the opposite of a wet mount. We have a wet mount, which is liquidy on the surface. We place a cover slip. The other method, the second method, is fixing it. So attaching the cells to the slide, which is often achieved by either heating it up, which is heat fixing, or chemically treating the specimen. So in addition to attaching the specimen on the slide, fixation also kills the microorganisms in the specimen. So in this case, then it would not be a living tissue. Unlike the wet mount, some wet mounts can be living tissue, so you can drop, for example, some type of specimen that's swimming around in some water or other solution on the wet mount and still look at them. In fixation, oftentimes this is killing the organism through either heat or through chemical, and it stops their movement and metabolism while preserving the integrity of their cellular components. So the positive is that we're stopping the movement, so if it's something that moves around a lot, we can see it, and it also preserves the integrity of the cell. On the other hand, if it's something that we want to see move around, then this is not a good option. To heat fix a sample, a thin layer of the specimen is spread on the slide. This is called a smear, and then the slide is briefly heated over a heat source. A lot of times this will be a Bunsen burner or an incinerator that's just passed not in the flame so much, but as above the flame, just to heat it up a little bit. Chemical fixatives are often preferable to heat for tissue specimens. Chemical agents such as acetic acid, ethanol, methanol, formaldehyde or formalin, and glutaraldehyde can denature proteins, stop biochemical reactions, and stabilize cell structures in tissue samples. So as far as chemical agents are concerned, or chemical fixatives, it depends on what it is that the person is looking for, what it is that they want to look at to use a certain chemical in order to fix the slide. So here you can see just kind of these pictures up here. A specimen can be heat fixed using a slide warmer. So in this case it's a slide warmer. You just set it here and it's heating it up a bit. It's going to heat it up. Here is an incinerator. So you're not putting it actually in the incinerator here, but here it's on the outside edge, you can see here. And then here, chemical fixation. So it's being placed into a fixative, in this case, the 10% formalin. In addition to fixation, staining is almost always applied. So staining is almost always applied to color certain features of the specimen before examining it. So stains or dyes contain salts made up of a positive ion and a negative ion. And then depending on the type of dye, the positive or the negative ion may be the chromophore. So remember the chromophore. For example, a natural chromophore was the chlorophyll in a plant. So if you put a plant on a slide and look at it through a light microscope, you can actually see it without having to stain it. But if you want to see other things, or if you want to see something that doesn't have chlorophyll in it, then you need to have a chromophore. So the positive or the negative ion may be the chromophore, which is the colored ion. The other, the uncolored ion, is called the counter ion. Okay, so in one situation, the positive would be the chromophore, and then the uncolored ion would be the counter ion, the negative. If the chromophore is the positively charged ion, there we go, the stain is classified as a basic dye. So if the chromophore, the one that does the staining, is positively charged, it's a basic dye. If the negative ion is the chromophore, then it's considered an acidic dye. So dyes are selected for staining based on the chemical properties of the dye and the specimen before being observed. So this will determine how the dye will interact with the specimen. So in most cases, it's preferable to use a positive stain, which is a dye that will be absorbed by the cells or the organisms being observed. So then it will add the color to the objects of interest and make them stand out against the background. So most times what we're doing is we're doing a positive stain. So you can see here, A, down here, the Bacillus anthracis cells, they have absorbed the crystal violet, which is a positive stain. So it's staining those organisms so you can really see them against the background. However, sometimes it's advantageous to use a negative stain, which is absorbed by the background, but not by the cells or the organisms in the specimen. So negative staining procedures produces an outline or silhouette of the organism with a colorful background. So we can kind of scroll up here and take a look at these. So here, like we said in A, Bacillus anthracis, they've absorbed the crystal violet, which is a basic positive stain. And then in B, this specimen, Spinoloricus, a microscopic marine organism, has been stained with rose bengal, which is a positive acidic stain. So this one's acidic and that one's positive. And then in C, the B megatherium appear to be white because they've not absorbed the negative red stain applied to the slide. So in this case, the red stain is in the background, and then we see the organism here is not stained. So because cells typically have negatively charged cell walls, the positive chromophores in the basic dyes tend to stick to the cell walls, making them positive stains. The commonly used basic dyes such as basic fuchsin, crystal violet, malachite green, methylene blue, and safranin typically serve as positive stains. And you should know these things, okay? As we're going through, it's important for you to know these different staining mechanisms, and we'll talk more about these as we're moving through the specific staining types. So you should know that these are basic dyes, okay? Positive stains, basic dyes, crystal violet, malachite green, methylene blue, and safranin are positive stains. On the other hand, the negatively charged chromophores and acidic dyes are repelled by the negatively charged cell walls, making them negative stains. So commonly used acidic dyes are acid fuchsin. Okay, so up here we have basic fuchsin. Down here we have acid fuchsin, eosin, and rose bengal. So some staining techniques involve the application of only one dye to the sample. Others require more than one dye. So in simple staining, a single dye is used to emphasize a particular structure in the specimen. So a simple stain will make all of the organisms in the sample appear to be the same color. So even if there's more than one type of organism. So if we're just talking about a simple stain, you put a dye on there, it's going to dye everything just to give it some contrast. So in that case, we're not talking about trying to determine what the organism is. It's just trying to determine if there is an organism on the slide. In contrast, we have differential staining, and that distinguishes organisms based on their interactions with multiple stains. So in other words, two organisms in a differentially stained sample may appear to be different colors. So in this case, when we're talking about a differential staining, it's to be able to determine what organisms are in there and look at the different organisms versus just saying, is there anything in this sample? Differential staining techniques commonly used include gram staining, acid-fast staining, endospore staining, Flagella staining, and capsule staining. Again, you should know those things and know that they are a differential stain and not just a simple stain. And we're going to talk about those in more detail now. So first we have our gram stain. The gram stain procedure is a differential staining procedure. We just said that. It involves multiple steps, and you need to know these multiple steps. It was developed by Hans Christian Gram in 1884, and it is an effective method to distinguish between bacteria with different types of cell walls. So even today it remains one of the most frequently used staining techniques. So this is incredibly common, and you should know these steps and know why it's used. So our gram staining, the first step here is we use crystal violet, and this is the primary stain. So this is applied to a heat-fixed smear, not a wet mount, but a heat-fixed smear, giving all of the cells a purple color. Okay, so first we apply crystal violet, that's the primary stain, and it makes all the cells purple in color. Then we add grams iodine, hence the gram staining, and this is a mordant. So the mordant for gram staining is grams iodine. And a mordant is a substance used to set or stabilize the stain or dye. So we're going to put the mordant on, and it's going to set that crystal violet in. Okay, it's going to stabilize it. So in this case, grams iodine acts like a trapping agent that complexes with the crystal violet, making the crystal violet iodine complex clump and stay contained in thick layers of peptidoglycan in the cell walls. Then we're going to add a decolorizing agent, usually ethanol or acetone ethanol solution. And then cells that have thick peptidoglycan layers in their cell walls are much less affected by the decolorizing agent. Okay, so we have put the primary stain on, the crystal violet, then we add our mordant, which is grams iodine, and then we have the grams iodine crystal violet complex is going to clump and stay inside very tightly thick peptidoglycan cell walls. Then we put this decolorizing agent on there, and it's going to wash away some of that crystal violet iodine complex. But the ones that are stuck, the ones that have thick peptidoglycan layers, will stay. So it's less affected by the decolorizing agent. So they will retain the crystal violet dye and remain purple. However, the decolorizing agent more easily washes the dye out of cells that have a thinner peptidoglycan layer, and then making them again colorless. So if there's a very thin layer of peptidoglycan, then it'll wash it out. If it's a thick layer, it stays purple. Then we have our secondary counter stain, and this is usually saffronin when we do gram staining. And then this stains the decolorized cells, the ones that had the gram stain or the crystal violet washed out. It stains those cells pink. Less noticeable in the cells that still contain the crystal violet. So it's going to stain everything pink, but the ones that have the crystal violet are still such a deep dark purple, you can't really tell that there's any pink added. So it's going to stain the other cells pink. So in this case then we end up with cells that have a thick peptidoglycan wall are purple from the crystal violet, and those that have a thin peptidoglycan wall are then pink. Here we have this all laid out here in a nice diagram. The step 1, step 2, step 3, step 4. If this is an easier way for you to visualize it, then that is fantastic. And then down here I'm going to finish talking about, I can probably put it about right here, finish talking about the gram stain and why we see what we see. So the purple, the crystal violet stain cells, are referred to as gram-positive cells. Okay, while the red safranin dyed cells are gram-negative. So when you're looking at a gram stain, those that stayed purple, the ones that had thick peptidoglycan wall, are called gram-positive, and the ones that have thin peptidoglycan wall are gram-negative. However, there are several important considerations in interpreting the results of a gram stain. So first, older bacterial cells may have damage to their cell walls that cause them to appear gram-negative, even if they are in fact gram-positive. Thus, it's best to use fresh bacterial cultures for gram staining, so we don't want them to be too old. We want to have nice fresh cultures. Second, errors such as leaving the decolorizer on too long can affect the results. So if we decolorize too long, it can wash away some of the purple. So even though it stays in there pretty well, if it's on for a very long time, it can wash away some of the purple, making it look kind of pink. So in some cases, most cells will appear gram-positive, while few appear gram-negative. So this suggests damage to the individual cells, or that the decolorizer was left on for too long. The cells should still be classified as gram-positive if they are all the same species rather than a mixed culture. So it's just important that while you're going through the staining procedure that you listen to the amount of time that you're supposed to do it, and make sure you're looking very closely at that so that you don't affect the cells to where it's difficult to figure out what's happening there. So besides their differing interactions with dyes and decolorizing agents, the chemical differences between the gram-positive and gram-negative cells have other implications with clinical relevance. So gram staining can help clinicians classify bacterial pathogens in a simple in a sample into categories associated with specific properties. So things that are gram-positive or gram-negative can be classified in that way. It's not just to say, okay, these pick up more purple, these pick up the pink, and you can see the differences. Gram-negative bacteria tend to be more resistant to certain antibiotics than gram-positive bacteria. So seeing that thinner peptidoglycan wall when you're doing something in the clinic, looking at these things, then they can say, okay, well, since this bacteria seems to be gram-negative, then we can administer this antibiotic. And we'll talk about that more in other chapters. And then that's up here in the chart also. So it mentions gram-positive here, picking up the purple, and then gram-negative. Okay. All right. So moving on to our acid-fast stains. So that was our gram stain. Now we're looking at the acid-fast stains. So acid-fast staining is another commonly used stain. It's a differential staining technique, again, not simple staining, but it differentiates between things. It can be an important diagnostic tool. So an acid-fast stain is able to differentiate two types of gram-positive cells. So if one first does a gram stain, you can figure out what is gram-positive, what is gram-negative, and then you can isolate that gram-positive and then use an acid-fast stain. Isolate what seems to be gram-positive, and then you can distinguish between different types of gram-positive cells. So it distinguishes between those that have a waxy mycolic acid in their cell walls and those that do not. Okay, so our gram stain looked at the thickness of the cell wall, the thickness of the peptidoglycan. Gram-positive had thicker peptidoglycan, gram-negative had thinner. In acid-fast stains, it can distinguish between waxy mycolic acids in their cell walls and those that do not have these waxy mycolic acids. Two different methods for acid-fast staining are the Zeal-Nielsen technique and the Kinyoun technique. Both use carbolfuchsin as the primary stain. The waxy acid-fast cells retain the carbolfuchsin even after a decolorizing agent, so an acid alcohol solution is applied. So again, kind of like the gram stain, we put carbolfuchsin on it. Then when we decolorize it, the ones that have the waxy acid-fast or the waxy mycolic acids in their cell walls keep that stain in it. And then we have the secondary counterstain, which is methylene blue, is applied, which then renders the non-acid-fast cells blue. So those that do not have that waxy mycolic acid. The fundamental difference between the two carbolfuchsin-based methods is whether heat is used during the primary staining process. So the Zeal-Nielsen method uses heat to infuse that carbolfuchsin into the acid-fast cells. The Canyun method does not use heat. So both techniques are used, are good diagnostic tools because there are a number of specific diseases caused by acid-fast bacteria, or AFB. They're present in a tissue sample. Their red or pink color can then be clearly seen against the blue background of the surrounding tissue. So that methylene blue stains everything else, and it makes it a very, very clear stain. We'll see here. So here, Mycobacterium tuberculosis here. This is an acid-fast stain. So the Mycobacterium tuberculosis cells are these bright red cells, very clear. And then the surrounding growth indicator is this blue because of the methylene blue that was added as the secondary stain or the counter stain. All right, next we have our capsule staining. So certain bacteria and yeast have a protective outer structure called a capsule, and we're going to talk more about that in future chapters, but they have this capsule. Bacteria or yeast can have a capsule. Since the presence of this capsule is related to the microbe's virulence, so its ability to cause disease, the ability to determine whether the cells in a sample have a capsule is an important diagnostic tool. So if somebody can determine, if a physician can determine if that bacteria or if that yeast has a capsule on it, then it can know, the person can know whether or not some sort of medication will work or whether it won't work, because it can tell how strong that bacteria or yeast is. Capsules do not absorb most basic dyes. Therefore, a negative staining technique, so staining around the cells, is typically used for capsule staining. So things like gram staining or acid-fast staining usually do not pick up the actual capsule on the cells. It may stain the cells, but not make a noticeable capsule to where you can determine that it has a capsule. So then we use something like a negative staining technique to do capsule staining. So the dye stains the background but does not penetrate the capsules, so then it appears like a halo around the cells, like halos around the border of the cells. And you can kind of see that. You can see this kind of halo effect around the cells. The specimen does not need to be heat fixed prior to negative staining. One common negative staining technique for yeast and bacteria that have capsules is to add a few drops of India ink or nigrosin to a specimen. So you add a couple of those drops, and then you can see whether or not you get this halo effect. So other capsular stains can also be used to negatively stain the encapsulated cells. Alternatively, positive and negative stain techniques can be combined to visualize capsules. So the positive stain colors the body of the cell, and then the negative stain colors the background but not the capsule, leaving a halo effect. So that's what we're seeing here. So in A, right here, they use the India ink to stain the background of the cells, the Cryptococcus neoformans. And so you can see that we have this background here where the stain is, the India ink, and then you can see the cells themselves are not stained. And you can see the halos around the cells here. And then in B, crystal violet and copper sulfate dyes cannot penetrate the bacillus cells in this negatively stained sample. So the encapsulated cells appear to have a light blue halo. Okay, so the crystal violet and the copper sulfate can't get into the bacillus cells. So you can see though that the cells are stained here, and then the background was stained here. So then you can see this nice white capsule surrounding it. All right. Next we have our endospore staining. So endospores are structures that are produced within certain bacterial cells that allow them to survive harsh conditions. So it's kind of like a capsule, except even stronger. It is going to allow them to, we're going to talk more about them in future chapters, but it allows them to stick around much longer. So they create these or make these endospores. So gram staining alone cannot be used to see endospores because they appear clear when gram stain cells are viewed. So you may see other aspects again when you gram stain, but you won't actually be able to pick up and see the endospores themselves. So endospore staining uses two stains to differentiate the endospores from the rest of the cell. So you can stain the cell and also the endospore and see the difference between the two. The Schaefer-Fulton method, which is the most common one, uses heat to push the primary stain, the malachite green, into the endospore. So our primary stain is malachite green, and that is going to stain. You can see it's pointing out these green endospores inside the bacterial cells. Washing it with water then decolorizes the cell, so it washes the malachite green out of the cell, but the endospore retains that green stain. The cell is then counterstained pink with safranin, and then the resulting image is like below here. You can see the shape and the location of the endospore here, the green part, if they are present, and then the green endospores will appear either within the pink vegetative cells or separate from the pink cells altogether. If there are no endospores present, then you will only see these pink cells. So you can see here's an endospore that is outside of the cell here. You can see others that are within the cells. And then if there were not any endospores, perhaps here, you can see there aren't any endospores in those bacteria, and they just are stained pink. All right, so endospore staining techniques are important for identifying Bacillus and Clostridium, two genera of endospore-producing bacteria that contain clinically significant species. So we're going to talk about different type of species and what they do. So right now it's just important that you understand that an endospore staining technique can identify a Bacillus species or a Clostridium species. So for example, B. anthracis, which causes anthrax, is of particular concern. Its spores can be used as a bioterrorism agent, so it's important to be able to do endospore staining because those can be used as bioterrorism. So also C. difficile is an important species responsible for the typical hospital-acquired infection known as C. diff or C. difficile. So it's important to know whether or not something has an endospore, so a physician can know what to do in order to try to prevent that illness or disease. Next we have Flagella staining. So flagella or flagellum is a tail-like cellular structure used for locomotion. So the flagella can whip around and allow the bacteria, archaea, and some eukaryotes to move. Because they are so thin, flagella typically cannot be seen under light microscope without a special stain. So you may do a gram stain, for example, but then you wouldn't actually pick up the flagella. So there are specific Flagella staining techniques. So the Flagella staining thickens the flagella by first applying a mordant. So remember a mordant, like in the gram stain. Generally tannic acid, but sometimes potassium alum, which coats the flagella, and then the specimen is stained with pararosaniline. Most commonly, or basic fuchsin. So you see here we have our mordant that is applied, it coats the flagella, then we add our stain, and then we can see our flagella. And you can see that here, all of these little kind of swirly guys that are coming up here, spiral guys, those are the flagella on those bacteria. So flagella stain of Bacillus cereus, common cause of foodborne illness, Bacillus cereus. Reveals that the cells have numerous flagella used for locomotion. So though flagella staining is uncommon in clinical settings, they most times are not concerned with whether or not there's a flagella. Its technique is used by microbiologists since the location and the number of flagella can be useful in classifying and identifying bacteria. So it's used most often in a laboratory so that people can classify the type of bacteria. When using this technique, it's important to handle the specimen with great care because flagella are delicate and they can be easily damaged or pulled off, and then you won't be able to actually see, locate, or count the number of flagella. So that's a little bit of a fragile one. The other ones you don't have to be as fragile with. It's of course good to be as fragile as possible so you don't burst any cells or wash away your stain, but that one you need to be particularly careful with. So here's a breakdown of some of our staining techniques, right? So we have our basic stains, acid stains, negative stains. These are some simple stains, right? Remember, a simple stain doesn't differentiate between different things. So we can just use some of these things we talked about as a simple stain just to stain everything on the slide. We can use a basic stain, an acidic stain, or a negative stain, or down here we can do differential stains. Remember, a differential stain is differentiating between different types of bacteria. So we talked about gram stain acid-fast stain, endospore stain. We talked about the Flagella stain, and we talked about the capsule stain. So you should know all of this information. This is a nice little chart you can use perhaps to study, to know the stain type, the specific dyes that are used, why it's used, the purpose, the outcome, what you get, and then you can see the sample images here. All right, so now we're going to switch from light microscopy to the electron microscopy and preparing a sample for visualization in an electron microscope. So samples to be analyzed using a TEM must be very thin. We talked about how you have to slice it very, very thin. But cells are often too soft to cut really, really thin. So even with diamond knives. So to cut cells without damage, the cells must be embedded in a plastic resin and then dehydrated through a series of soaks in ethanol solution. So it gets more and more increasing the percentage of ethanol, which is drying out those cells. So they embed it in plastic and then they dehydrate it using ethanol. The ethanol then is going to replace the water in the cells, and the resin dissolves in the ethanol and enters the cell where it solidifies. So the resin is going to dissolve in the ethanol, and it's going to enter the cell just because the ethanol replaces the water in the cells, and then it's going to solidify in there. So basically it's kind of turning the cells into plastic into a resin. Next, the thin sections are cut using a specialized device called an ultramicrotome. So an ultramicrotome is going to get these incredibly, incredibly thin slices. Then finally, the samples are fixed to fine copper wire or carbon fiber grids and then stained, not with our colored dyes, but with substances like uranyl acetate or osmium tetroxide, which contain electron-dense heavy metal atoms. So remember, we did talk about that when we talked about the microscopes, how they're stained with heavy metals, because then the electrons, as they bounce through and against these things, react differently. All right, so the TEM, remember, it has to be really, really thin. Okay, so we have the resin. It's embedded in this plastic resin and dehydrated using ethanol. The ethanol is going to dissolve the resin and replace the water in the cells that then solidifies, makes it a nice solid structure, and then they cut this into very thin sections. And then when they cut it into thin sections after that, they fix it to this copper wire or carbon fiber grid and then stain it with heavy metals. When samples are prepared for SEM, then they must also be dehydrated using the ethanol series. However, they must be even drier than the TEM. So the critical point drying with inert liquid carbon dioxide under pressure is used to displace the water from the specimen. So they need to be even more dehydrated. So they're dehydrated using the ethanol series, and then they do critical point drying with the inert liquid carbon dioxide that's under pressure, and it displaces the water from the specimen. So after drying then, the specimens are sputter coated with metal, again using the metal by knocking atoms off of a palladium target with energetic particles. So they use a palladium target and then they hit that with energetic particles, and it then sputter coats the specimen that has been incredibly dried out. This sputter coating then prevents specimens from becoming charged by the SEM's electron beam. All right, so just remember those specific things. So just kind of this preparation process, something we won't do in the class. We don't have electron microscopes, of course, but the TEM and then the SEM. All right, so then preparation and staining for other microscopes. Remember, we had those probe microscopes that we talked about and 2-photon microscopes. So here's some other staining methods here. So samples for the fluorescence, remember we talked about fluorescence microscopes and confocal microscopy, are prepared similar to samples for the light microscopes, except that the dyes are fluorochromes. So remember, we use fluorochromes in the fluorescence microscopy. Stains are often diluted in liquid before applying to the slide. So some dyes attach to an antibody to stain specific proteins on specific types of cells, which in that case is immunofluorescence. So cells have specific antibodies on them. Mention that briefly, meaning self or non-self, and then those specific antibodies have those fluorochromes attached to them. So remember the primary and the secondary when we were talking about the fluorescence microscopy. So some dyes attach to the antibody to stain specific proteins on the specific type of cells, which is called immunofluorescence. Others may attach to DNA molecules in the process called fluorescence in situ hybridization. So that's FISH for short, FISH. Causing the cells to be stained based on whether they have a specific DNA sequence. So there can be a specific DNA sequence that a physician or a person in a laboratory is looking for, and then they can use the fluorochromes to attach to that very specific sequence. They can get that attached, and then they can look at it underneath that microscope, and whichever ones are glowing with the fluorescence, with the fluorochromes, are going to be the ones that have that specific DNA sequence in them. Sample preparation for 2-photon microscopy is similar to the fluorescence microscopy, except for the use of infrared dyes. So remember that one included infrared. So specimens for STM need to be on a very clean, anatomically smooth surface. They are often mica coated with gold. Toluene vapor is a common fixative. All right. A little less specific on those. So then we have, again, these things at the end, our clinical focus, ion ethics, and then we have our summary. So if you are studying and looking through this material, again, the summary is very helpful at the end of the chapter, and then there are the review questions that are also very helpful. So I definitely suggest going through those things as you're studying and going through the chapter. So make sure that you know, going back up here, just this last section, make sure you know how to prepare slides for fluorescence and confocal and for the 2-photon. Remember how to prepare slides for the electron microscopy, TEM and SEM. And then these are very, very helpful charts. So you should know the information that's contained within them, the stain type, the dyes that are used, the purpose, outcome, know what they are supposed to look like, understand what a simple stain is compared to a differential stain and different types, so basic stain, negative stain, acidic stain, the dyes, the purpose, the outcome, and those types of things. So understand the difference between the different types of staining like endospore, flagella, capsule, and then going all the way back to the beginning, you should understand the difference between a wet mount versus a fixed mount here. All right, so then we're going to move on to parts of Chapter 3 next.